All right, so today is kind of the beginning of the semester, the beginning of the rest of the semester, I should say. So far, we've really just kind of built up to the point where we are at this spot now. We're really just kind of getting into what the body is made up of, which are cells, which cells are made up of various organelles, providing various functions. If you can work all the way back even to atoms and molecules, that wonderful stuff that we talked about. Really just kind of prepared us to talk about systems the rest of the semester, which a system is a group of organs, and together they perform special functions. Here on our page here, we can see the various organs and tissues of the nervous system. In this lecture, we're going to be talking about those structures and we'll look at their functions a little bit. We'll get into that a little bit more in greater detail. But I kind of don't follow the book in the order that the book goes for a couple of reasons. The first system that the book talks about is the integumentary system, which we really don't even get into in this class. Same thing for the skeletal system. Those systems are more kind of anatomically based stuff. And we'll talk a little bit about those as we kind of progress through other systems with their relationship together. But then they go from the skeletal system into muscles and the muscular system. Learning about muscles is great and all, and we will do that. But to me, it makes the most sense to talk about the nervous system and the endocrine systems first. Okay, the nervous system encompasses 4 different lecture packets, 3 of which will be included in this unit. And then the endocrine system is one huge lecture that will be in our next unit after we've talked about the nervous system. Reason is because, remember, those 2 systems are in charge of everything else, responsible for maintaining homeostasis. And so I like to talk about those because, when we get to like muscles, we're already going to know what an action potential is. So that's important. Without an action potential, muscles won't contract. So that's kind of why I kind of steer a different way than the book does. All right. Kind of the first thing we're going to do today is talk about the organization and the structures that make up the nervous system. You can see here on the first page, we've got a figure with 2 boxes and some structures labeled and located within those boxes. The first one over there on the left includes the brain and spinal cord, which are components of what we call the CNS. And then over on the right for the PNS, we've got cranial nerves, spinal nerves, and sensory receptors in the skin. There's not a whole lot of structures that make up the nervous system. It's really one of the least amount of organs for all the systems of the body. With that said, still probably the most complex. Our brain is the major structure making up our nervous system. Within our brain are 10 billion neurons, or somewhere around that number. That's an incredible amount. We'll talk about what neurons are today. Associated with our brain are 12 pairs, so 24 total cranial nerves. These guys will send signals to and from the brain. They do things like bring the sensation of taste into our brain, sensation of vision, hearing. Those are all special senses, and they are going to travel their signals by way of our cranial nerves. There's a pair of cranial nerves that actually go from our brain all the way down into our abdomen, doing a lot of different things that relate to our various organs. They're located in our thoracic and abdominal regions. Extending off of the brain down through our vertebral canal, connecting our brain to the spinal nerves is a singular spinal cord, very complicated tissue that we'll talk a little bit about today and more so in our upcoming lectures. There are 31 pairs, so 62 total spinal nerves that are the major nerves of the nervous system, making up and or serving the left and right sides of the body. Associated with our brain and or our cranial and our spinal nerves are ganglia, which is this kind of massive nervous tissue, primarily the cell bodies. We'll talk about what a cell body is here today of a neuron. Then we've got enteric plexuses that regulate the digestive system. We'll talk a little bit about the enteric nervous system in this lecture, but we'll get into that a little bit in greater detail when we actually talk about the digestive system, because Obviously, digestive system is what the entire nervous system controls and helps to regulate. So it makes sense that we'll talk about those later this semester. And then we've already actually talked about sensoreceptors since the very first lecture of the class when we talked about action, or not action potentials, but feedback. Remember we talked about sensors and receptors. Those are the same things that detect changes. That can be internal or external. And sensory receptors can either be a part of a neuron or specialized cells that interpret these external, internal stimuli. Okay? And sensory receptors actually provide us with our first overall function of the nervous system, which is the sensory function. Now this page that describes the three major general functions of the nervous system really kind of just describes the feedback loop, which we should know really well by now since you've already been tested over it. Remember the first component of a feedback loop were those sensors or receptors. Okay. That detected a change. Internal, external, doesn't matter what we're talking about. Okay. Those sensory receptors, sensors or receptors, a lot of times are neurons. The type of neurons are sensory or afferent. We'll talk about what those words and what type of neurons these are today. Okay. Sensory neurons provide sensory function. That's about as easy as it could ever be. Detecting changes like a blood acidity inside of our body, or a raindrop hitting your arm, or like today where it's frigid outside and you are interpreting that signal really quickly. Now, when I say interpret that signal, that's taking place inside of our brain generally, which is where we perform our integrative function, okay, where we process that sensory information, store some of it, make decisions for appropriate responses. Like in the case where it's cold outside, we know that immediately because we have perception over a stimulus like super cold. Okay? Perception is our conscious awareness of sensory stimuli. When I say conscious awareness, it means that we are totally aware of it. Totally aware that it's frigid outside within a matter of seconds. Do we have conscious awareness of all sensory stimuli? So we're consciously aware of everything changing in our body all the time? No. Do we consciously know when our pH is dropped from 7.4 to 7.38? No, we might be consciously aware of effects resulting from changes like that, like when our blood sugar drops and we start to feel like crap. But we don't necessarily know, oh crap, my blood sugar dropped 2 millimeters or 2 milligrams per deciliter. Or my blood pressure went up 2 millimeters of mercury. We're not always consciously aware of those more internal stimuli changes. Okay? Now, perception is great. We perceive a lot of things. You right now are perceiving my talk, my words. If I were to start singing, you would be able to perceive my change in voice and recognize that I'm not just speaking, I'm singing. So music awareness, vision, smell, taste. And we're always going to be sensory aware of those things. And that's because of the high-level functions that occur in our brain. Okay. Now, our spinal cord can be our integrating center in some cases, performing this integrative function. But most of the time, it's our brain. The type of neurons that make up our brain and our spinal cord primarily are called association or interneurons. Okay. I prefer interneurons. You'll see why in a little bit. Okay, but interneurons perform the integrative function, which is nice because they both begin with INT. Motor function is performed by motor neurons or efferent neurons, which is how we respond to that original stimulus. Just like we talked about with feedback, you send a signal in telling your body that it's cold, your brain processes that, your brain sends out an output signal. That output signal is coming by way of these motor neurons performing the motor function. And the motor function means that we're going to activate or inhibit, in some cases, effectors, which will be muscles and glands, skeletal muscle, cardiac muscle, smooth muscle. Glands could be internal, like endocrine glands, or external, like sweat glands. And this is controlled by way of the cranial and spinal nerves, sending the signals outward from the brain spinal cord. Okay? So, like I said, this is basically just a revamp of what we talked about with feedback. Now, that first page showed the two things that were part of the CNS and the other stuff that was part of the PNS. Guess what CNS and PNS are short for? Central nervous system and peripheral nervous system. So if you need to write those abbreviations out, might not be a bad idea, because we will use those quite a bit. The CNS, or central nervous system, is a structural and functional center of the entire nervous system, and this is your brain and spinal cord. Those are the two parts of the central nervous system. This is where we perform that integration, processing that input, dictating out the correct and appropriate outputs. Okay, so this is our command center, primarily done within the brain. Okay. Not always your spinal cord is going to be the command center for some reflexes. Like when you touch something hot, we don't have to send that signal up to our brain, process it, decide what to do. Your spinal cord processes and decides what to do so we can respond quicker. Okay. Peripheral nervous system is going to basically be everything else. So everything in the periphery or outer regions of the nervous system, which includes the spinal cord, the cranial nerves carrying signals to and from the brain, spinal nerves carrying signals to and from the spinal cord, the ganglia, which are associated with those two different structures, and then those sensory receptors, which are either going to be the dendrites of sensory neurons, which we'll talk about what dendrites are, and then in our next units we'll talk more about the sensory nervous system and the specialized cells that make it up, like in the case of, in our mouth for our tongue, in our eyes, our ears, and so on and so forth. Okay. There is literally a lecture where we spend the majority of our time talking about sensory receptors and our ability to perform that sensory function. Okay. Our next lecture is on the central nervous system, where we'll talk about functions of the brain, different parts of the brain. We'll talk about the different functions and parts of the spinal cords. Okay, the third function, or the third lecture of this unit, is where we'll talk about the divisions of the peripheral nervous system, which, because there are so many parts to it, we can subdivide it. And we do so based on who's being regulated. So the effectors. Okay, we have on this page the somatic and autonomic nervous systems, which are the two parts of the peripheral nervous system. The key with somatic is that this is associated with voluntary control, and we're carrying signals to and from skeletal muscle. If you have to underline voluntary and skeletal muscle, highlight it, star it, whatever you need to do, that's very important to know for somatic. So when I wave my hand at you guys, I'm utilizing the somatic division of my peripheral nervous system. And who's doing the waving or moving of my fingers? It's the skeletal muscles in my hand. And I'm consciously aware and doing this under my own control. Okay? I'm purposely doing this, or I'm purposely not doing it. Okay, that's what we're talking about with voluntary. Okay? What we talk about with somatic is mainly motor or output, but there's also sensory or input. So as it states here, the somatic nervous system consists of sensory neurons conveying info from somatic receptors on our head, body wall, limbs, as well as the receptors for the special senses. We'll talk more about those in our next unit. Okay, for this unit, in our last lecture of this unit, we'll talk mainly about the motor aspect, signals going out to the skeletal muscle. Okay. Now, next to autonomic, I always tell students, if it helps, write out the word automatic, because the words sound very similar and it describes the activity of the autonomic, which is involuntary, meaning that we don't have to have conscious awareness and I don't have to use my brain power to perform it. Okay. I don't have to tell my stomach to release enzymes. I don't have to tell my heart to beat harder. Don't have to tell the smooth muscle in my blood vessels to dilate or contract. So we're talking about just automatically done for us. The effectors in an autonomic nervous system reflex are going to be our cardiac muscle in our heart, smooth muscle in places like our blood vessels, and then a lot of our glands, which can be those like the endocrine glands, thyroid, parathyroid, or as simple as our sweat glands, which, when it's hot, we don't have to sit there and go, okay, it's hot, I need to sweat. It's just automatically done for us. Now, adipose or fat tissue, as well as other tissues, can be controlled by the autonomic nervous system, but it's these first three that are the primary effectors. Now, we can then subdivide the autonomic nervous system into three parts: a sympathetic, a parasympathetic, and then the third is the enteric. Of the three, we don't talk very much about the enteric. We'll talk mainly about sympathetic and parasympathetic. The sympathetic produces a response that's coined as fight or flight, okay, which describes something that we do in response to like a scary situation, okay, where you either need to fight whatever the issue is or get the heck away from it, like a bear attacking you. Okay. What we do in a fight or flight response is adapt the body to perform physical activity, or to exercise, or to run or fight, which are both examples of a exercise situation. It does everything to get us jacked up, basically. So like an adrenaline rush is what we're talking about here. Your heart rate's going to go way high. It's going to beat really hard. Feels like your heart's beating out of your chest. Your blood pressure is going to go up. Your sweat's going to be produced. Your blood vessels are dilated in some places, constricted in other places. Your pupils are dilated. Your bronchial tree is dilated. We're just doing everything we can to fight off whatever's going on or run away from it. Okay. The opposite of that is the rest and digest response, which is produced by the parasympathetic nervous system, which does everything to get your body, in a resting condition. Okay. Releasing saliva, digestive enzymes into your stomach. Right now, this one's what's active in you guys. Okay. Your heart rate's low, your blood pressure should be low, your breathing rate's pretty low. If I told you guys, put your stuff away, we're going to take the test on this unit today, in your last 30 minutes of class, what's going to happen? You're going to start sweating, your heart rate's going to go up, some of you might want to run out of the room, some of you might want to fight me. So that can illustrate just how quickly we can turn on a dime from a rest and digest to a fight or flight response. So these two systems are like a teeter-totter. One goes up, the other one goes down, vice versa. Okay. Now the third division of the autonomic nervous system is the enteric, which is specific to the GI tract. Which the GI tract or gastrointestinal tract includes our our stomach and our intestines. So both the small and large intestines. Like I said, we'll talk mostly about this later in the semester when we get to digestion. And what's cool is many of the neurons of the enteric nervous system function independently of the autonomic and central nervous systems, but they're also going to communicate with the central nervous system by way of that sympathetic parasympathetic aspect. Okay. Think about this. Rest and digest, produced by the parasympathetic division, is going to promote the action of the enteric. But what about in a fight or flight response? Do you think we're caring about digesting that cheeseburger we had for lunch? Or are we going to devote all of our resources to getting away from the bear? So as the sympathetic nervous system becomes active, the enteric nervous system will become inhibited. When the parasympathetic is active, the enteric will also be active. Okay. There is sensory and motor to the enteric. We'll talk about sensory neurons with regards to GI tract changes like stretch, pH, like when your stomach is filling up with food, that stomach will stretch. There are stretch-sensitive receptors that detect that stretch and will turn on by way of motor output. Contraction of the muscle in your stomach to start to mix up the food in our digestive enzymes. Will release chemicals. So it's pretty cool how that all relates. Both the PNS and the CNS include cells that form incoming and outgoing pathways. We've already seen these 4 words: afferent, sensory, motor, efferent. Those were all described with regard to neurons performing those various nervous system functions. The afferent division is always incoming information. The other name for afferent is sensory, which means the signals are going towards the brain or spinal cord, which can either come from somatic senses, special senses. We will talk all about this stuff in the first part of our next unit. We will talk all about the motor, efferent, outgoing information in the third lecture of this unit, which will compare somatic and autonomic in greater detail. Somatic, which we said is voluntary control of skeletal muscle. Autonomic, which is automatic or involuntary control. It involves sympathetic, parasympathetic that control our smooth muscle, our cardiac muscle, and our glands. The enteric, which it controls our GI tract. Okay. Notice the direction of the arrows: blue going towards the CNS, red going away from. So, not that you need to know this table or figure, whatever you want to call it, flow chart, but it's kind of neat to show how it's all put together. What we're going to do now is move away from talking about components of the nervous system and the way that it's organized, to talking about the cells that make up the nervous system. There are 2 major types of cells that make up the nervous system: neurons, which we've already mentioned quite a bit, and then supporting cells, which we call neuroglia or glial cells, which we're getting ready to talk about right now. Of the 2 types, we'll talk in much more great detail about neurons. But neurons, which are the cells that signal to and from, back and forth in our body, won't function correctly without the help of these supporting cells. We describe them as non-excitable, which means that they don't send signals, so they're not the nerve signal guys. They're the supporting cells that help that process to take place. There are 6 types of neuroglia. 4 of them are found specific to the CNS. And our CNS is once again what? Central nervous system, and includes our brain and spinal cord. Okay. Here on the next page, we can see the 4 that are specific to the CNS. Okay. We can also see neurons, which we'll talk about here in a little bit. First type are called astrocytes. There's 2 different types. We're not really concerned about the differences in those. But they help primarily to wrap around blood vessels in our brain and spinal cord and to form what's called the blood-brain barrier, or BBB. They will pick up nutrients from our blood and pass it along to the delicate cells of our brain, to the neurons. Okay. They prevent toxic stuff, some drugs and that kind of stuff, from leaving the blood and entering into our brain and spinal cord, which would inhibit nerve signaling. Okay. Now you can see they actually will support neurons as well, so they have that kind of secondary function as well. Microglial cells are these very small cells. These are phagocytes in our brain and spinal cord. Remember, phagocytes perform phagocytosis, which is like Pac-Man running around chewing stuff up. Okay. They evoke immune responses in areas of infection, trauma, or stroke, removing cell debris, microbes, and damaged nervous tissue by phagocytosis, eating that stuff up. Ependymal cells were the ones down here on the bottom. We find these in areas of our brain called ventricles, the central canal of our spinal cord, the places where we produce and circulate cerebrospinal fluid. We've talked about cerebrospinal fluid since that first lecture. This is a very important fluid. Without the correct concentration of cerebrospinal fluid, our neurons would not work. And the cilia help to circulate it, the microvilli help to release it. Okay. And so that's the overall function of ependymal cells. Produce positive motor and assist in the circulation of the CSF. Oligodendrocytes are probably the coolest ones for their name. They look like an octopus. They wrap around the axon of a neuron. Axon is this long wire and form what we call a myelin sheath. This is a fatty insulation that helps speed up nerve signaling. Okay. Now, singular oligodendrocytes will wrap around multiple axons. So you can see here it's wrapping around the axon of this neuron. It's also wrapping around the axon of this neuron. Okay. Here's another one, this neuron, and this neuron. Notice that this kind of pinkish shaded myelin sheath is not continuous all the way down the length of the axon. There are these little blank gaps called nodes of Ranvier. Okay, and we'll talk about those here a little bit more in detail after we talk about the other two neuroglia. The other two neuroglia are found specific to the peripheral nervous system, which hence the name PNS neuroglia. That includes Schwann cells and satellite cells. I don't have a picture of these, but that's okay. Schwann cells are just like oligodendrocytes in that they form the myelin sheath, okay, which, as I mentioned, speeds up nerve signaling, but also protects the nerve fiber, so that axon, insulates them from one another. The difference between these guys and oligodendrocytes is that these guys only wrap around, well, I don't want to say that because I don't want confusion. What is something that's cool about these guys, so let me backtrack, is that they have this outer wrapping called a neurolemma, which is helpful for regenerating damaged nerve fibers, so axons that have become damaged. Much more so than what can happen in the central nervous system, because this is common knowledge, but if you have brain or spinal cord damage, is that usually very reparable? No, generally that's permanent, right? Okay. In the peripheral nervous system, the Schwann cells help to fix that. Okay. As I mentioned, just like in our central nervous system, the Schwann cells, so these aren't Schwann cells, but the same thing will be occurring. The wrapping, that myelin sheath, is not continuous. We are left with these nodes of Ranvier, which allows a nerve signal to skip from node to node like a rock skipping on water. We call this process saltatory conduction, and we'll talk about it on Wednesday, but it's super fast. It's much faster than if this axon was left uncovered, which some axons are. Okay? So sometimes it is completely devoid of any myelin. Satellite cells are also associated with neurons, specifically their cell body, which is this part of a neuron. They wrap around the cell body of neurons, help to regulate exchanges of materials between the cell bodies and the interstitial fluid. Okay. Before we move on to neurons, I want to just last talk about gray and white matter, which does involve neurons. Some of you guys have taken anatomy and have gotten to dissect sheep brains. Some of you guys will get to do that this semester. One of the coolest things about dissecting the brain or dissecting the spinal cord is that you will find parts of the brain and spinal cord that are more light in color, part of them that are more kind of dark or dull color. These are what we call white and gray matter within the brain and spinal cord, and it's due to the presence or absence of myelin. If it is white in color, it is due to neurons that have lots and lots of myelin. Okay? Because of the fatty makeup of myelin, it's going to look really white. In our brain, it's going to form the inside, covered by a shell of gray matter. On the spinal cord is going to be on the outside with the gray matter on the inside, so they're opposite. Okay. Most of our nerves in our peripheral nervous system will be white matter. In our spinal cord and our brain, we call the white matter tracts, and we'll talk all about tracts later this week. Okay. The unmyelinated fibers, which have very little to no myelin, form this gray matter. It forms an H in our inner spinal cord and a shell in our brain. Okay? We'll talk all about this shell in our brain. Very important. We call it the cerebral cortex. That's where we do a lot of that perception that I was talking about earlier. Okay? This is where you find the cell bodies of neurons. They're dendrites, the axon terminals, or in some cases just bundles of unmyelinated axons in neuroglia. Okay? And like I said, it's gray in color due to very little or no myelin. Really, that's about it for the supporting cells. We will mention them a few more times, but the rest of this lecture will be really mainly about the neurons, which are the major cells of the nervous system, providing the nervous system with those three major functions, as we saw earlier today. We describe neurons as excitable cells, which means that they can rapidly change their membrane potential. You guys remember what resting membrane potential was? It's that negative charge on the inside of cells due to unequal numbers of ions and other charged particles. Remember what the resting membrane potential number was for most cells? Negative 60. Okay, it's going to be negative 70 for neurons, actually. Okay. When a neuron moves ions in and out, it changes that resting membrane potential. That's how we conduct impulses. And impulses are going to make possible all those nervous system functions. So therefore, we can think of neurons as like the wiring of our body. Okay. Here on the next page, we can see, this is a very simplistic view of a neuron, but it is good enough to show us the major parts, which includes the cell body, which is the control center of the neuron. This is where you find most of the major organelles that we've talked about recently. Here is a big old nucleus. You'll find mitochondria. You'll find an intricate arrangement of those cytoskeleton structures. You'll find lysosomes. There's Golgi. There's RAR, which gets a special name called Nissl bodies. So that's the nucleus. Okay. Or that's the cell body, excuse me. Associated with the cell body, I should keep saying nucleus. The cell body are these extensions, which we collectively call nerve fibers. Okay. There's two different types of nerve fibers. Branches coming off of the cell body, generally a whole bunch of them, like tree branches, we call dendrites, and then generally a long axon, okay, which is like the wire. Okay. Dendrites, as it states back here on the previous page, are the primary sites for receiving signals from other neurons. Okay. And so a signal will stimulate the dendrites, the signal is then processed by the cell body. The axons, which begins at this tapering portion called the axon hillock, are responsible for taking that signal and sending it down the length of the axon from the cell body to a remote spot. Axons can be super long, okay? Like the sensory neurons that are found in like your toes. Okay, those axons run from basically your toes all the way into your spinal cord. So these cells can be super long, specifically the axons, several feet in many cases. Okay. That's why here they're showing this little gap. That's not an actual gap. They're just cutting down the length because it's not up to, or it's not to scale. Most of the time, axons are pretty unbranched. There will be some cases where there is an axon collateral where they don't show it, but there would be this associated with it, carrying the signal not only this way, but this way as well. At the end of the axon will be some more branches. We call these axon terminals, or telodendria. If you zoom in at the end of them, you'll see that they have a swollen synaptic end bulb or synaptic end knob, where we find bubbles called synaptic vesicles full of chemicals called neurotransmitters, which we'll talk about those in great detail on Wednesday and possibly Friday, because that's kind of towards the end of the lecture. It's pretty cool what happens. The signal starts here and travels down to here, and then something special happens there. Okay. The signal that begins back up here, it's not labeled here, but somewhere right in here in the axon hillock is called the trigger zone. This is exactly what it sounds like. That's where the nerve signal is first triggered. Okay. And then it is like lighting a fuse. It's going to burn the whole way down. And it does so super, super amazingly fast. Now, what was pictured there on the previous page was one of our three structural neurons. And that was actually a multipolar neuron, the most common type. We've got two other types, unipolar and bipolar. These names come for how many extensions come off of the cell body. I've drawn some simple pictures of them over here on the board. Okay. Multipolar neurons have at least 3 extensions. Here's the cell body. Here's a couple of dendrites. There's probably a whole bunch more than that. And 1 axon. Okay. These guys make up something like 90% of the neurons of our body. Okay. The neurons in our brain, this is mainly the type that they are. Okay. Unipolar, got the cell body off to the side with a singular extension that forks to go to some axon terminals on one end and the dendrites on the other. Okay. Bipolar, these are the most rare. We find these only in places like our eye and our ear. They've got the cell body in between 2 wires. One goes to the dendrites, one goes to the axon terminals. Okay. Now, down below these, I've listed their functional classification. Which describes the direction in which they send signals. This is how we had the neurons listed back when we described their functions, or the functions of the nervous system. The sensory function was performed by sensory or afferent neurons. The integrative function was performed by interneurons or association neurons. The motor function was performed by motor neurons or efferent neurons. It all relates to the direction of travel. Travel of the nerve impulse, that is. Sensory or afferent neurons are generally going to be, or most of the time going to be, unipolar in structure. And they're going to go towards the brain and spinal cord, carrying signals like, pressure or touch, vibration, itch, tickle. Okay. Now, bipolar will also be sensory in function, and they carry signals like, hearing or smell into our brain. Okay. Our brain and spinal cord are primarily made up of interneurons. You guys remember what the prefix inter means? Between. That's why I like the name interneurons, because these are located between sensory and motor neurons. Here's our sensory unipolar neuron. Carries a signal, doesn't matter what it is, maybe it's touch, maybe it's itch, maybe it's tickle, whatever, into our brain or spinal cord. Look at the top there, it says central nervous system. Okay. This is where we perform integration by way of these interneurons, which happen to be multipolar in structure, as it states over there on the board. Also multipolar in structure are our motor or efferent neurons, which are the neurons that carry the signal out of the brain and spinal cord to the effectors. And that should illustrate why multipolar neurons are the most common type, because they are what make up our brain, and they also are the ones that stimulate our muscles to contract, our sweat to be produced by sweat glands, etc., etc. Now, functionally, the most common type of neurons are interneurons, because those are the ones that make up our brain and spinal cord, which are the primary structures of the body. What do we say, 10 billion neurons in our brain? They're going to be that type. Multipolar interneurons. Doesn't matter the type of neuron, they all have the capacity to communicate. And it's by electrical excitability, which means that they can flow ions in or out, changing that resting membrane potential. The two types of signals include graded or local, used only for short distance communication. Action potentials, which are much more complicated, allowing for communication over long distances within the body. Okay. We're not going to get to those today, but we'll talk about both of those on Wednesday. In living cells, the flow of ions constitutes electrical current flowing down neurons. Graded potentials and action potentials occur because membranes of neurons contain many different kinds of ion channels, opening or closing in response to specific stimulus. Here's a pretty complex looking illustration of just how amazing the nervous system is with performing something as simple as writing your notes on your page. Okay. 8 steps involved in this simple process. Simple with air quotes. The graded potential is going to develop when your fingers first touch a pen. That happens in a sensory neuron in your fingers. That then becomes an action potential that spreads into your spinal cord. There's our sensory neuron, by the way. Into our spinal cord, and then up the spinal cord, we perform a synapse, which is where communication occurs between one neuron and another neuron. That signal is then going to create an action potential up into our thalamus in our brain. Another synapse, so signaling between two neurons, all the way up to our cortex. That's our highest brain center, where we perform perception. Then we have the motor aspect of things. The signal comes down by an action potential through a motor neuron, down our brain, into our spinal cord, all the way down and out of our spinal cord by way of a lower motor neuron that then activates an action potential in our muscles. And this causes them to contract, allowing you to write with your pen. And all this happens within no time. Think about how amazing that is. You're really going to appreciate this when we learn more about action potentials next time. We learned about resting membrane potential last week. The excessive negative charge in the cytosol of a resting or unstimulated cell. We said for most cells it was negative 60. In neurons, it's a little more negative. It's negative 70. Talk about why that is here in a second. There are 4 ion channels that can open or close here listed, which would then change that resting membrane potential, which is important for neuron signaling. Okay. The first type are called leakage or non-gated. We can see a picture of them here on the next page. These basically just randomly open or close, and when they do, they're going to allow the ion to move down its gradient. Okay, like potassium leakage channel here, that opens up and would allow potassium out because there's more potassium in than out. Okay. We don't talk about those very much at all. We will talk a lot about ligand-gated channels, which require a chemical to bind to it before it opens. That chemical is generally going to be a neurotransmitter, a hormone, or it could be another ion. And the chemical, like you see here with this little red kind of diamond looking like structure, acetylcholine is who it is, kind of works like a key going into a door lock, which then unlocks the door to open up the door. Okay. We'll talk a lot about those in our next couple days. Mechanically gated, these are pretty cool. Some type of mechanical stimulus opens them up. Okay, it could be like a sound wave, could be a touch, pressure, or even a tissue stretching. We don't talk a whole lot about those, but that's how we can detect a stretch in our muscles. Voltage gated channels open in response to a change in membrane potential or voltage. Okay, these are very, very important for action potentials. We'll talk about those in great detail next time. As you can see here with our voltage-gated potassium channel, that's closed at negative 70. But when the voltage is negative 50, they are open. Okay. Down at the bottom are the table, or is a table with those same ions listed. Okay, so you can kind of read through that on your own if you want. Okay. One more slide, and then I'll let you guys free. The reason that our resting membrane potential is negative 70 is because of that small buildup of negative ions in the cytosol along the inside of the membrane, and equal buildup of positive on the the outside. 3 major factors contribute to this. There is an unequal distribution of ions in the ECF and cytosol. The ECF, or extracellular space outside of the cell, is rich with sodium chloride. The inside is full of potassium. Okay. Well, there's also a lot of negatively charged phosphates, like the 3 phosphates that are attached to each ATP, okay, and even proteins. Now, neurons have more potassium leak channels than sodium leak channels. So more potassium can exit than sodium can enter. You're losing more positive out than gaining positive in. Okay. Most anions, what charge does an anion have? Negative. So those negative charged ions can't leave the cell because they're kind of stuck to a molecule that won't diffuse out. And then finally, that sodium-potassium ATPase pump, which pumps 3 positives out, 2 positives in. That's a result loss of 1 positive charge, isn't it? All that contributes to this negative 70 inside the cell, which is important for what we'll talk about next time, which are the 2 different types of potentials, graded or local, and action potentials. And so we'll pick up with that first thing.